Okay, this video is an introduction to the Karyotyping Lab. We are going to be showing you some karyotypes, but before we show you a karyotype, we want to talk about how you get the cells to make a karyotype. So we do what's called an amniocentesis, where we have to get a cell sample from around a fetus. Now, if we want to look at chromosomes, first of all, a fetus is usually where we would want to get those cells because we're often looking for genetic chromosomal abnormalities, and we want to find out before a baby is born if they have extra chromosomes or are missing chromosomes. But it's also really a great place to study mitosis because we would have a lot of very rapid cell division. But the way that we get cells from near a fetus is called an amniocentesis, and it's on the screen in blue. And they take a very large needle and puncture the abdomen and into the amniotic sac and get a few cells from the fluid that the baby is floating in. They don't actually get it directly from the baby. So this is a kind of picture of a chromosome. These aren't photographs of real chromosomes. This is sort of like a computer-generated simulation. But basically, you would get a bunch of chromosomes scattered all over the place. And now they probably have computers that pair this up, but the way we used to do this is we used to give you a sheet like this and have you cut them out and match them all up. Now there are 3 physical characteristics that you would use to match them up. If you notice, there are large ones and small ones. So you would use size, you would use the banding pattern when they're stained, and you would use the location of the centromeres. Some of them have centromeres right in the middle, some of them have them off center, some of them have it right at the end. So again, you're looking at size, banding pattern, and location of the centromere. The centromeres, if you recall, are the part of the chromosome or the area where the 2 replicated chromosomes are held together, but that's the unreplicated portion. So there is a homologous pair that has been circled for you in green. There's another homologous pair that's been circled for you in red. And we had to label the X and the Y chromosomes because they don't match. So it would be really hard for you guys to have figured out where the X and the Y chromosome. The X is significantly larger than the Y chromosome. So this is the X chromosome and that's a Y chromosome. So in this diagram, they're all placed together. So we basically would get some cells and grow them in a tissue culture and look for one that's in metaphase and make what's called a metaphase smear, and then take a photograph and enlarge it and organize the homologous pairs by size. So if you look at this, this is a human karyotype, and there are 23 pairs, and they're arranged in size order from largest to the smallest, except for the sex-determining chromosomes at the end. So here is a homologous pair, here is a homologous pair. So these are actual photographs of real human chromosomes. So if you have 2 X chromosomes, the notation is 46XX, 46 because that's a number of chromosomes, XX tells you you have 2 X chromosomes, and in that case, the baby would be a girl. And if the 23rd pair is one larger and one smaller, this is 1 X chromosomes and 1 Y, it is a boy. And since females only have X chromosomes, it's actually the father that determines whether the baby will be a male or female. So the female, all of her eggs will have an X chromosome in them. Sperm, half of them will have an X and the other half will have a Y chromosome. So any cell that has homologous pairs is considered diploid. So if you look at this picture, you can see 2 large chromosomes and 2 smaller chromosomes. And the homologous pairs, one came from the father and one came from the mother of this person. So if this was a cell in your body, you could say the light blue ones came from your mother and the dark blue ones came from your father. You would call this cell diploid because it has 2 of each type of chromosome. The prefix di means 2. And you would also know that it is a body cell. Another name for body cell is somatic cell. And the very first cell that is diploid that became you is called a zygote, and that's what a fertilized egg is called. So when the nucleus of a sperm joins the nucleus of an egg, we have fertilization, and the fertilized egg is called a zygote. Zygotes are diploid. And then they start dividing mitotically to grow. And so every cell after that would be diploid. In fact, all cells are diploid except for gametes, and we'll talk about those next. So cells that contain only one of each homologous chromosome are called haploid or monoploid. Haploid is the more common term that I have heard, but monoploid also is accurate. And cells that are haploid are notated with an N rather than the 2N the diploid cells were. And in human, these sperm and egg cells are formed in testes and the ovaries, and they're known as gametes. So they're known as gametes in every species, but eggs and sperm are what we call them in human and in many other species too. And the scientific name for egg is ova. O-V-A is plural, and O-V-U-M, ovum, is singular. So if you look at this cell, we have one chromosome that's smaller and the other one that's larger. And so they're not in homologous pairs. And so there's only 2 chromosomes in this species. Now, meiosis produces gametes. So the production of gametes is called meiosis. It's also sometimes referred to as reduction division because it reduces the number of chromosomes exactly in half, so that when fertilization takes place, you don't have double the number of chromosomes. If no reduction happened in chromosome number before fertilization, every generation would have double the number of chromosomes, and that would be a problem. Having even one extra chromosome can cause problems for humans, so having whole extra sets would be really problematic. It's actually quite common for in the plant kingdom for there to be what we call polyploid cells and polyploid organisms where they have multiple copies of chromosomes. And that's not actually a very big deal, but it is in humans. It's not something that's very safe. So that is the end of the introduction to the Meiosis Lab.